Thank you. Good morning, everyone. So I'm very happy to introduce our next guest. He's a very good collaborator and a close friend of us. Professor Elwira SIENIAWSKA from the Medical University of Lublin in Poland. Dr. SIENIAWSKA received her PhD in pharmaceutical sciences from Medical University of Lublin in 2012. She currently is a professor, congrats Elwira, at the Department of Natural Products, Chemistry at Medical University of Lublin in Poland, and her scientific experience is based on phytochemistry. She worked on isolation and utilization of components of essential oils as antimicrobials and boosters of antimicrobial drugs activity. However, the liquid chromatography mass spectrometry analysis of bacterial metabolites produced under stress conditions is her current interest. During her research, she completed 3-month scholarships in the Department of Chemistry, Biotechnology and Food Science in Norwegian Institute of Life Sciences, published more than 80 scientific papers and several book chapters, participated in numerous scientific conferences relevant to natural products, and obtained 6 Polish patents. Since 2011, she served as a principal investigator and co-investigator of research projects and supervised numerous students. So Elwira, please, you have the virtual floor.
Thank you. Thank you, Professor Firascu. It's my great pleasure to be here with you in this hybrid form. I am very happy. It's my second participation in this very successful conference, chemistry-oriented, although my talk will be more biology-oriented, bioactivity-oriented. However, without chemistry, all these observations which I would like to present to you would not be possible to have. So the topic of my today's speech is observations about natural products, how they alter metabolic pathways in mycobacteria, and what I would like to summarize here. These are results from since 2018. So last 3 or 4 years, we were performing studies which were aimed to check how different natural products interact with mycobacteria, tuberculosis, and how they can change their metabolic pathways. And we wanted to check these very active natural products, because they were able to inhibit bacteria growth. So if we are going into natural products, we all know that screening of antimicrobial is still ongoing. We still need some new compounds. No one denies it. And since many active products has their leads from nature, so compounds from plants, from fungi, from also other bacteria, from endophytic bacteria, or from lichens, these are lead compounds. There are so many active compounds, numerous of these compounds. However, as we can imagine, they all act as antimicrobials in different manners. And what we would like to see, these antimicrobial compounds are targeting bacteria in quite few ways. So here we can see that I'm sorry for this rectangles. I don't know how to delete it. Okay, I will just ignore it. So the 5 or 6 main ways in which numerous natural compounds and also antibiotics target bacteria in general. These are related to ATP biosynthesis, also cell envelope formation and development, DNA replication, transcription, and translation. And in general, all these processes in a bacteria cell consume a lot of energy. And just to underline that protein biosynthesis consumes about 70% of ATP generation. So it means when we target natural with natural product and with antibiotics, when we target bacteria, it is important to observe metabolic changes, and these metabolic changes very frequently are related to metabolic adaptation connected to energy generation and energy consumption. And in this way, I would like to underline why Mycobacterium tuberculosis is an interesting subject. Mycobacterium tuberculosis is a slow-growing organism. It means that it replicates very slowly, once per 24 hours. In general, bacteria replicate, for example, like Escherichia coli every 20 minutes. So to obtain larger biomass, we should harvest bacteria for 3 weeks. However, it's not my point that we should be patient. My point is that when we observe metabolic changes, and when these metabolic changes are slower, we are more likely to do not miss any important observations. So that's why Mycobacterium tuberculosis is a target which is worth to study from the point of view. However, these observations can be also confirmed in other bacteria. When we are talking about Mycobacterium tuberculosis, this is bacteria which requires for its growth a high pressure of oxygen, because it's because of its physiology. It targets lungs of human, of course, and high pressure of oxygen, its aerobic bacteria, is needed for its growth. So it was shown that bactericidal antibiotics in Mycobacteria impact bacteria in the presence of oxygen more likely. It means that in the presence of oxygen, antibiotics cause production of reactive oxygen species, which destroy macromolecules in the bacterial cells. This can be related to the cell wall, but also directly to proteins and lipids. So this was also confirmed by the addition of Elwira, which can scavenge oxygen species, and in this way protect from the oxidative damage caused by bactericidal antibiotics. So this was observation done in Mycobacterium tuberculosis. Also, it was shown to more precisely define what is this oxidative damage, in which way oxidative damage is performed. So it was shown that it relates to oxidation of deoxycytidine triphosphate in Mycobacteria, and it was observed importantly not only after one antibiotic, but after different antibiotics like ciprofloxacin, rifampicin, and streptomycin. And it is worth to say, underline that 3 of these antibiotics have different targets in mycobacterial cells. So it means that there are some other general pathways which are targeted in bacteria. Not only primary drug target is the only one way in which bacteria suffer under the stress conditions. These observations were also confirmed in other rapidly growing bacteria. And maybe also to underline what is rapidly growing. So if we see bacteria colonies in the for example, in the flask in shorter time than 1 week, so we say that these are rapidly growing bacteria. In case of Mycobacterium tuberculosis, we need more than 7 days to have these observations. And when we are speaking about mechanism of action, because the aim of observations of in the metabolic activity of bacteria under stress conditions, under natural product conditions, is to determine the mechanism of action. So we can say that these two general mechanisms can be observed. So bactericidal antibiotics, and also we have bacteriostatic antibiotics. In case of bactericidal antibiotics, when in different manners, but for example, this oxidative damage occurs, it causes damage to intracellular macromolecules, as I mentioned before. So it causes elevated strength response, which can be observed, as we also did in our research, by the increase in the activity of transcription factors, which transfer the stress signal from outside the cell into the cell and also interact with each other to maintain homeostasis and counteract the stress conditions. So this also involves increased metabolic activity, because if some damage is caused, so increased metabolic activity is aiming to restore balance and, for example, rebuild damaged structures. So as a consequence, depletion of the cellular energy resources occurs. So it means that when a cell increases development of new structures, of course, it loses energy pools. And finally, the toxic metabolic byproducts are accumulated in the cells after all these events, which cause cell death. So this is mechanism of bactericidal antibiotics. When we speak about bacteriostatic agents, we observe something opposite. So we observe decreased cellular respiration. So it means that energy is not used to release it in the high extent. So in the cell, we still can observe accumulated resources. Reduced macromolecular biosynthesis and slowing metabolic activity results in the cell stasis. So bacteria stop to grow, but they are not killed, so can perform again increased metabolic activity after the removal of stressing agent. So these observations in our laboratory are done by untargeted metabolomic research, and this you can see experimental metabolomic workflow, and this all needs chemistry as a base, because we start firstly with experimental design, which of course involves all the establishing parameters which we would like to study and just check within bacteria. I mean, for example, the period of times in which we make stop for metabolism and make extraction. Also the ways of performing extraction, which finally ends with different metabolites which are present in the extract, for example, lipophilic, hydrophilic. Also, we can extract different layers of the mycobacteria cells. So it also provides information about the changes in the different parts of the bacterial cell. And we need chemistry, because we perform analysis involving liquid chromatography mass spectrometry. We also experimented with nuclear magnetic resonance. But if you, for example, have accessible liquid gas chromatography mass spectrometry, you can focus on volatile compounds and also have this observation in this range of compounds. So after analysis, we obtained a lot of data, which should be computational processed, and it is worth to mention that in the metabolomic studies, we need biological replicates, so at least 6 to 10 biological samples are needed. So we should normalize them, perform peak detection, and also peak grouping, which is usually performed by software. And we go to statistical data analysis. So additional visualization of our data is obtained when we simply cluster results for control and test groups, and we compare if we have any differences or not. Finally, we come to biological interpretation, which is also performed by statistical tools, which are widely freely available through the internet, but also we have a range of subscription option also in the internet. However, we can start with this easy software to perform, for example, enrichment analysis or interactions between genes and metabolites. So I would like to present you a few examples of our research which were related to different natural products. And my focus is to show you how really different natural products interact with mycobacteria, what is common in these interactions. So it means which metabolic pathways were altered by all these compounds on majority of these compounds. So it means that it is really not easy to find one molecular target in the bacteria cell, which is a perfect hit. And for example, as we studied usnic acid, that this usnic acid interact only with one protein, and we have a perfect drug which hits only one enzyme in Mycobacterium and nothing more. No, so if it was proven that even in case of antibiotics we have secondary targets, so we can also expect that in case of natural products we can some general response. And if we come out to results, we observed two major metabolic responses, which were firstly related to upregulation in the lipid molecules needed in the formation of a cell wall. And here is the structure of the Mycobacterium tuberculosis cell wall, which is very thick and very characteristic because it has numerous layers. And coming out from the upper layer, it is the capsule, which is formed from non-covalently molecules. Then we have mecolic acid, which are very long fatty acids. And then we come to the polymers of sugars and proteins, which are arabinogalactan and lipomannan. And finally, at the bottom, we have the cell membrane. The other mechanism was related to activation of the detoxification mechanism and reactive species scavengers. And these two mechanisms were, let's say, general and characteristic for all studied natural compounds. So in case of structure of a cell envelope, we observed that increased synthesis in the layer of outer membrane, which is composed of mecolic acid and also glucose monomecolates and thiocerol dimethylcerosates. This was increased, thickened in case of cinnamon essential oil and also in case of usnic acid. Also, cinnamon essential oil caused increased synthesis of precursors of lipomannan and lipoarabinomannan. These lipid molecules are very characteristic and needed for a sealing of the pore in the and cavities in the cell wall, but also these structures have some immunological importance because it is a factor of virulence. If it comes to cell membrane, we observed increased number of molecules which are needed for a formation of a cell membrane. These were phosphoinositol-based molecules, but phosphodiacylglycerol phosphoethanolamines, diacylglycerols, and these acylated and mannosilated forms. And this layer was increased after propolis treatment and usnic acid treatment. When we observed also more closely the composition of the acylated and mannosilated forms of inositol, we saw after treatment with bacteria treatment with cinnamon essential oil that increased number was characteristic, which was related to restoring of membrane fluidity, which is when membrane is more rigid, it is more susceptible to the leaking, and when membrane is fluid, it's more flexible and provides better protection. Also, we observed some interesting results in case of treatment bacteria with Tanchinions. It was interesting that almost all molecules were removed from inside layers of Mycobacterium envelope to the outside layers. So we observed a lot of molecules in the culture medium, meaning that it was some kind of damage in the cell envelope, and the molecules were secreted or just released to the culture medium. So it was a disruption caused by Tanchinions. So also at this layer of cell envelope, these mechanisms were taking place. And in case of detoxification mechanism, we observed that in case that cinnamon essential oil and usnic acid increased production or also not only the production of factor 220, which is required for xenobiotic in general metabolism, but also the reduced form of this factor, which was in case of cinnamon essential oil treatment, and this particular reduced form is needed for xenobiotic metabolism and neutralization of these xenobiotics. I'm sorry, not this way. And the other observation was the flavin metabolism was increased, and this is very important because the riboflavin and in general flavins are important cofactors needed for redox reactions, and these redox reactions are important for a production of numerous molecules in the cell. So it means not only for the detoxification, but also for the production of peptides, mecolic acids, lipids, and DNA molecules. So redox reactions are very important for, in general, increased metabolic activity. The other observation related to redox homeostasis was increased in the sulfur-containing amino acids after the treatment with Nepalese propolis, and redox reactions which involve coenzymes like nicotinate and nicotinamide precursors. I would like to underline what was just the specific observation. So I just mentioned it, that it was a disruption of bacterial cell envelope surface, and this was particularly observed for Tanchinions. Besides, the other observations were typical for all studied natural products, all studied extract. So it means that it's not easy to target Mycobacteria in one point, but in general observations, we have similar observation like for other natural compounds and also for antibiotics. And here as a conclusion, I would like to also show you an example that how we can deduce bactericidal mechanism of action. For example, usnic acid caused also hydrolysis of diacylglycerols and trioxyroglycerols. In this way, free fatty acids are liberated, and this fatty acid are needed for beta oxidation and also for the production of numerous compounds which are important in the increased metabolic activity when not only energy should be restored, but also the structure of it should be restored. So it needs to the depletion of the energy resource and toxic metabolic byproducts, and finally cell death. And we also observed elevated stress response, which was measured by sigma factors, increased expression of these factors indicate that stress response was activated, that bacteria tried to overcome the stressing agent, and we also correlated this particular sigma factors with metabolic activity. It is not the subject of this lecture here, but it helped also to to deduce the more specific response of bacteria to the stressing agents. And it is also worth to underline that the combination not only of metabolomics but also other omics like transcriptomics is important to have a full, more wide, let's say, view on the metabolic changes in bacteria, not also in Mycobacterium tuberculosis. At the end, I would like to again to thank you for this possibility to share my research with you. Also to underline that chemistry is a base of everything for, of course, of life, and we use it every day. I would like to acknowledge also my collaborators from Department of Biochemistry and Biotechnology from Medical University of Lublin, and also from Stefan Nagel of Institute of Microbiology from Bulgarian Academy of Sciences. And I would also like to thank you for your kind attention and invitation here. Thank you.
Thank you very much, Elwira. Now, if there are any questions from the room or from the online? No, we don't have any question. It was a very solid presentation, Elwira. Actually, we worked together for a couple of years now, and it's not a surprise for me, your presentation. You have very nice results.
Thank you.
We thank you for your participation today, and I do hope that next year we will be able to meet in person, hopefully at PRIOCHEM or at the workshop dedicated to the young students in the summer, our next chem workshop. And thank you once again.
Thank you, and good luck for next presentations today and tomorrow also.
Thank you.